Hello, my name is Dr. Claudia Chiocchini. I'm a staff scientist in R&D with molecular biology at Thermo Fisher Scientific. We are here to introduce a white paper titled Evaluation of GeneArt Gibson Assembly EX Cloning Technology to Build Large and Complex Assemblies. Building DNA constructs that are large in nature and contain no extraneous sequences is often the cornerstone of synthetic biology. One of the key engineering tools in constructing such DNA is Gibson assembly cloning, and we will discuss how the GeneArt Gibson Assembly EX cloning technology can be used to build very large constructs using 10 inserts along with cloning strategies. The success rate of fragment assembly is influenced by two main factors: the size and the number of fragments. The efficiency of Gibson assembly technology is generally more affected by an increasing number of fragments in a reaction than by increasing fragment size. Therefore, when building larger molecules, it is recommended to keep the number of fragments as small as possible. In this example, we assembled a 10 kb insert in a PUC19L vector using 5 2 kb fragments sharing 30 base pair overlaps. The assembly product from the GeneArt Gibson Assembly EX cloning kit is a covalently bonded DNA molecule that can be transformed into chemically competent or electrocompetent cells. High cloning efficiency between 95% and 100% was obtained in each case. There are several important considerations when assembling very large DNA constructs. One is that it is preferable to keep the number of fragments as low as possible and use longer overlaps, 50 to 100 base pair, to maximize the cloning efficiency. Another important consideration is the use of the correct cloning vector. E. coli typically displays a low tolerance for high copy plasmids containing large DNA constructs. To avoid this, it is recommended to use low copy plasmids that allow for propagation of the full-length construct without hampering the E. coli DNA replication cycle. Gibson assembly products are covalently bonded and can be therefore transformed by electroporation into electrocompetent bacterial cells. This increases transformation and cloning efficiency, and is strongly recommended when building large constructs. Here we show the design strategy for building a 25 kb construct using 2 inserts and a 50 kb construct using 4 inserts, sharing a 50 base pair overlap along with their respective cloning efficiencies. These were cloned into the low-copy PASE101 vector and transformed via electroporation into Invitrogen Electromox DH10B E. coli competent cells. 16 colonies were analyzed to calculate cloning efficiency. While cloning efficiency was expectedly lower, high colony numbers made it possible to identify a colony containing the full-length insert in both cases. This white paper explores the challenges and main factors to consider when building very large constructs. We demonstrate how GeneArt Gibson Assembly EX technology delivers high colony efficiency, both when joining numerous short fragments and when joining a few large fragments, thus offering maximal flexibility. If you would like to learn more about this topic, please visit thermofisher.com/Gibson Assembly.